Thank you for joining the Center for Systems Vision Science International Seminar Series supported by the Creating Future program of Ritsumeikan University through the grant from program to supporting research activity of female researchers MEX. This is the first special talk of this seminar series. It is my great honor to introduce the speaker of first talk, Dr. Steve DeVries. Dr. DeVries is the David Shock Professor of Ophthalmology and Physiology at Northwestern University Feinberg School of Medicine. Dr. DeVries received his MD and PhD from a combined degree program at the University of Chicago, where he worked with Dr. Eric Schwartz on the modulation of horizontal cell combined by coupling by dopamine. Dr. DeVries then joined Dr. Dennis Vera's laboratory at Stanford University for postdoc, where he worked on a newly developed retinal multi-electrode MEA recording technique. In 1998, Dr. DeVries joined the Visual Science Group at the University of Texas at Houston as an assistant professor and began his work on cone synapse. In 2001, Dr. DeVries moved to Northwestern University, where he is now a professor of ophthalmology and physiology and has combined his work on and continued his work on both the cone synapse and dichromatic color vision and inner retinal circuit. In 2018, in recognition of his long-term contribution to the field, Dr. DeVries received the Brian Boycott Award of FASEM meeting on retinal neurobiology and visual processing. Today, Dr. DeVries will talk about his latest work. The title is Design of the Mammalian Cone Photoreceptor Synapse. I heard his recent hobby is playing with Vibe, the virtual reality goggle, to go deep inside his new 3D super resolution images of the cone synapse. He will share some of that work, but not the 3 virtual reality experience yet in today's talk. Thank you very much, Dr. DeVries, for your talk as a first talk of our seminar series, and I really am looking forward to hearing your lecture.
Hi, everyone. I hope you can see my screen. Thank you, Dr. Koike, for that very nice introduction. I will be showing some super resolution images today, but unfortunately, we can't share in the virtual reality just yet. So I want to thank the Center for Systems Visual Science for sponsoring the talk and you for inviting me. I wish I could visit this year. But unfortunately, circumstances prevent that. However, hopefully this Zoom talk will be the next best thing. So today I'm going to talk about the design of the mammalian cone photoreceptor synapse. And of course, the cone photoreceptor synapse is a crucial link in the visual chain between phototransduction and perception in the visual cortex. It's also an important locus neurobiologically. It's a very intricate synapse where a single presynaptic neuron, a cone photoreceptor, transfers its signal to more than a dozen types of postsynaptic neurons. So it's kind of a unique synapse where a lot of processing is taking place. The cone synapse is also important from the point of view of medicine, with the ongoing push to perform photoreceptor transplantation. We look forward in the near future to being able to integrate photoreceptors into the remaining retina. And hopefully part of my work will shed light on the proper and appropriate wiring that one can expect in the optimal case. So today I want to talk about some fundamental principles of signaling at the cone photoreceptor synapse. One fundamental principle in particular, and I want to see how close functionally the different types of off-bipolar cell contexts come to meeting this sort of fundamental optimum. And the idea is very simply that a cone photoreceptor has many ribbons, on the order of 20 ribbon synapses, and signaling should be optimal when a bipolar cell samples the release from all of the ribbons. I'll explain theoretically why that makes sense, And then we'll go through physiologically to show you that that appears to be the case for the off bipolar cells, albeit in some unique ways for the different types. With that introduction, I'll move to the first slide. And again, the basic idea is that the estimate of the cone signal improves with the number of ribbons sampled by a bipolar cell. And this idea is not new to me. It actually takes off from work of Simon Laughlin in the 1990s on fly photoreceptors, demonstrating such a sampling optimum. But the fly synapse is composed of many discrete synaptic ribbons and is anatomically unlike the mammalian cone photoreceptor synapse, where ribbons are aggregated fairly close together. So we wonder whether the same sort of optimization holds true in mammalian cone photoreceptors. And so let me describe that optimization briefly. First, a cone photoreceptor in the mammalian retina contacts more than a dozen different types of bipolar cells. And contacting all these bipolar cells leads to a very complex wiring in the terminal. We understand a great deal about that wiring, and I'll just review that for you in a moment. From ultrastructural studies, the middle panel here shows a section of the primate cone terminal. This was kindly given to me by Peter Sterling, and I only learned later that this is Dr. Tsukamoto's work, very beautiful work, but I should have known that from the start. But this illustrates a cross-section of the primate cone terminal, showing the two types of ultrastructural contacts that the cone photoreceptor makes with postsynaptic cells, particularly the bipolar cells. And it starts to illustrate the very intricate nature of wiring at this synapse. One of the types of contacts is called an invagination. And at the apex of an invagination is a synaptic ribbon. And these are sites of calcium-dependent vesicle fusion and glutamate release. The components of the invagination are two horizontal cell processes which enter and terminate laterally. We won't be saying anything about horizontal cells today. And a central process which belongs to an invaginating bipolar cell, which is typically an on-type of bipolar cell, but in the ground squirrel, the cone-dominant retina species where I work, there's one type of off bipolar cell that makes an invaginating contact. And we'll look at that type of off bipolar cell. The other types of contacts are basal contacts, and there are very many basal contacts across the cone surface. And at these contacts, the membrane of the cone and bipolar cell come into close apposition, but we believe there's very little, if any, transmitter release at this location. Rather, my own work, among others, shows that it is actually glutamate diffusion from the ribbon sites in the invagination that supplies these postsynaptic contacts with their transmitter. And I should say that I'll be talking solely today about off bipolar cells. The off bipolar cells express ionotropic glutamate receptors. The cone terminal is a very compact synaptic surface. This is shown in the illustration at the bottom here. It's about 5 microns in diameter. The business end, where all the contacts occur, is really about 3 microns in diameter. And in terms of the ground squirrel, very similar in primate and mouse, there are about 20 ribbons spaced within the terminal. And here's the point I want to make in this introduction. Our current understanding is that vesicle fusion at ribbons occurs randomly under conditions of steady voltage. So to illustrate that idea, cones sit at a steady voltage in the dark of about minus 40 millivolts. And under those conditions, calcium channels are open and calcium can flow in continuously. And these ribbons are thought to act like conveyor belts. The vesicles at the bottom near the calcium channels and membrane fuse, releasing their contents into the synaptic cleft. And then they're replaced by new vesicles from further up the ribbon, or sometimes from the cytoplasm, which take a few moments to become competent to fuse. And we've animated this process here, showing the fusion at these white vesicles, which then fade out and are replaced by other vesicles up the chain. all right, which then prime. And I'm just going to take you through the animation here. Okay, occasionally we see two vesicles fuse, and that's multi-quantal release where a calcium channel opens below and then causes two adjacent vesicles to fuse. The point here is that under steady voltage conditions, the vesicles approach the membrane, they fuse, and they're randomly replaced by vesicles in time that become competent to fuse. So that release at a given ribbon occurs randomly. And indeed, there is no coordination between ribbons, as there is no or very little coordination between sites within ribbons. So if you're a postsynaptic bipolar cell and you're looking at the fusion that occurs, vesicle fusion that occurs at one ribbon, what you see are individual events separated in time, sometimes multi-quantal events, with these variable, sometimes fairly long intervals. And I'll put a number on that in a moment. So if you're a bipolar cell and you're just sampling a single invagination, the signal has quite a bit of noise. Over a long period of time, you can have an average mean rate, but if you break that period of time down into intervals, that rate will vary statistically. You can improve your ability to estimate the cone signal by sampling from additional ribbons. Indeed, you can improve it by sampling from all the ribbons. And the idea is as follows. When you sample from multiple ribbons, let's say 4 ribbons, your signal is 4 times as large. The signal variance is 4 times as large. But the important measure of the noise is not the variance, but the standard deviation. And that only goes up as the square root of the number of independent release sites sampled. So as you sample more ribbons, your signal to noise ratio improves. For example, sampling 4 ribbons gives you 4 times the signal of sampling 1 ribbon, but only twice the amount of noise. So conceptually, there is a drive toward wanting to sample from as many ribbons as possible, potentially all of the ribbons in the cone synapse, in order to obtain the best estimate of the cone signal. So that's sort of the fundamental concept, and we're going to use physiology to actually see that this sort of sampling, this sort of complete sampling occurs, at least for off bipolar cells. And this just shows an example here of what this sort of sampling may look like in terms of the contact of a single off bipolar cell. Okay, I need to make a point about a presynaptic vesicle release because the rate of release matters for this idea. If there's an extremely high rate of vesicle turnover at ribbons, then there'll really be a uniform concentration of glutamate across the terminal. And that glutamate concentration may ebb and flow like the tides or the waves in the sea, but all you need is 1 foot in the ocean at the beach in order to see how much the tide has come up. You don't need a diversity of contacts to measure tidal flow at the synapse. And tidal flow, as it turns out, can occur during an abrupt stimulus that is able to release all of the vesicles, but under steady conditions, our physiological results, capacitance measurements, and electron microscopic measurements of the cone terminal done by Chad Grabner in the lab, suggest that release is more discrete across the surface of the synapse in space and time. And I just want to put some numbers on this. This shows a diagram of a single, one of the 20 synaptic ribbons in a ground squirrel cone terminal. There are about 30 vesicles docked in the bottom row of each ribbon, 600 vesicles altogether. And for each ribbon, when you put it under maximal steady release conditions, the rate of turnover is 30 vesicles per second. We've modeled the effect of vesicle fusion in the synaptic cleft, and the glutamate concentration in the cleft after a vesicle fuses is only elevated for about a millisecond. So at a rate of 30 vesicles per second, there are going to be large gaps in the signal from any single invagination. For instance, a 30 millisecond gap is enough to contain a minimal cone response in this species, which takes about 10 milliseconds to peak and another 15 milliseconds to decay. Our modeling studies suggest not only that glutamate is there and gone within a millisecond, but that the concentrations of glutamate are attenuated by diffusion such that they're too low at distances more than halfway between invaginations to activate ionotropic glutamate receptors. So the picture that comes from these studies is that there'll be a vesicle released at one ribbon under steady conditions, a vesicle released at another, and a vesicle released at another. But very rarely, statistically, are the contents of two vesicles going to overlap to produce a gradient. And in this context, it makes sense to think about the sampling by the different types of bipolar cells and trying to see how well the different bipolar cells sample all of the ribbons in a cone terminal. There's an early hint about this sampling. Again, we work in the cone-dominant retina of the ground squirrel, this guy over here. We work on off-bipolar cells. There are 5 types of off-bipolar cells. We're just going to consider them in 3 broad categories: the CB1 cells, the CB3 cells, and the CB2 cells. Within a category, the properties are very similar. You don't have to memorize this. I'm going to go through the talk and use a little cartoon in each slide where it's relevant. But what really inspired us to think about sampling was some of the early confocal results obtained by Wei Li when he was a postdoc in the lab and published in 2006, where we imaged using a confocal microscope the contacts between a cone and the different types of bipolar cells. And you can see a diversity of contacts suggestive of the diversity of sampling. CB1A cells here make a single contact right in the center of the cone terminal. It's a basal contact. CB3 cells make multiple contacts, also basal, at the cone terminal. And CB2 cells make multiple contacts, invaginating. And we're going to consider for each of these 3 types of contacts how well they approximate this optimum of sampling all of the ribbons at the cone synapse. So we measure transmission at the cone synapse by making slices of the ground squirrel retina and using 2 pipettes to record simultaneously in the slice from a presynaptic cone and a postsynaptic bipolar cell. This is a totally light-adapted preparation. We're not using light signals at all. With the cone pipette, we control, whole cell pipette, we control the voltage of the cone and can evoke transmitter release at will. And with a pipette attached to the postsynaptic bipolar cell, we can record the response to the release transmitter. A typical response is shown below here. Typically, we'll hold the cone at -70 millivolts, a voltage at which the calcium channels are closed and all of those vesicles are docked at the cone synapse. And then we step to a very depolarized voltage of 0 millivolts, where all of those channels open very quickly and the vesicles fuse in mass. Glutamate enters the synaptic cleft. It diffuses to the receptors on the postsynaptic side. It opens those receptors with voltage clamping the bipolar cell at -70 millivolts. There is a large inward current due to the massive release of glutamate, and then that current decays to a much smaller steady level, consistent with the continuous turnover of ribbons at the synapse at that relatively low rate that I described. Now, in the experiments I'll describe today, we actually dialed down the stimulus from this very powerful stimulus and created a weaker stimulus for the cone that evokes, that engages more of the statistical nature of release. And we're going to make use of the statistical nature of release to examine the sampling at the synapse. And this just shows the responses of a postsynaptic bipolar cell, consecutive responses showing that follow a brief depolarization, 1 millisecond from -70 to about -30 millivolts in the cone. You can see a small response in the first trial, no response in the second 2, a much larger response in the 4th, and a response similar to the first one in the 5th trial. And I want to call your attention to these response failures. And the normal interpretation at a central synapse, or even the neuromuscular junction, of such a response failure is that the presynaptic cell is actually failing to release transmitter. So that these failures tell you something about presynaptic release. But we can't make that assumption at the cone photoreceptor synapse because this particular bipolar cell may be only sampling from a few of the ribbons at the synapse, and transmitter release may be occurring relatively far away at other ribbons. So indeed, during these failures, the cone could be releasing transmitter. Well, how do we solve this problem? Well, there's a very nice technique we can use at the cone synapse where we can record these synaptic events postsynaptically, but also at the same time from the cone photoreceptor, actually monitor the number of vesicles that have been released. And this trick, this technique makes use of a trick and a phenomenon first noted by Frank Werbelin's lab in the 90s, that cone photoreceptors express a glutamate transporter in their terminal. These show two cone photoreceptors outlined with PSD95. Ribeye is the synaptic release protein. EAT5 is the transporter. You can see there's a lot of transporters at these contacts. And so in this trick, we make use of the transporters to tell us when the photoreceptor has released a vesicle. And briefly, we do this as the idea behind this is as follows. The transporters not only mediate a voltage and sodium-dependent glutamate uptake, reuptake into the cone terminal, but they also contain an anion conductance, a channel that opens as long as glutamate is bound to the transporter. We fill the patch pipette with thiocyanate, which is a very permeable anion through this transporter, so that when the channel is open, we see a relatively large current. And through this means, whenever a vesicle at a ribbon fuses and releases its glutamate into the cleft, we can locally see the effect by monitoring the transporter current. We know we're looking at transporter currents because there's a specific antagonist to the transporter current, TBOA. Here we've evoked a large current with a brief depolarization at the cone synapse, recording the feedback onto the transporter, shown here under control conditions, and then in the presence of TBOA, the blocker. I want to note that these transporter currents are very prolonged, but we know that glutamate is high in the cleft only for a brief moment. It's actually the transporter which tightly binds glutamate that actually turns over at a very slow rate, about 10 per second, and remains active as long as glutamate is bound. That's very useful for us because it produces very large, slow currents that are easy to measure, but it does interfere if we want to look at the temporal aspect of glutamate release. But we're confining ourselves to a single impulse in these studies. So we can apply a series of depolarizations to the cone. We can record from the cone, and we can see a series of postsynaptic events. Here, color-coded to help you in terms of amplitude. There are two failures in the sequence, a number of cases where there's a detection of a single release event in the cone, 2 release events, 3 release events, 4 release events, and something that doesn't fit the Poisson distribution of release, which is likely to be a large multi-quantal release event. From such events, it's a pretty easy task to identify the unitary event, which helps us count the number of vesicles that have been released. And these just show that you can record spontaneous events in the cone photoreceptor. They're large, they're countable, they're very distinct. Many of them have a similar amplitude. And there are occasionally these multi-quantal events. Okay, so we can record events postsynaptically in the bipolar cell. We can count events presynaptically in the cone. How does this tell us about the sampling by the different bipolar cell types? So I want to start off by talking about the CB2 cell. And the CB2 cell is a bipolar cell, off-bipolar cell that makes invaginating contacts. And it may contact a portion, or perhaps even all, but more likely a portion of the invaginations in the cone terminal. And our idea about the sampling of release is that whenever a single vesicle, as small as a single vesicle, is released by a ribbon within an invagination, If there is a finger from the bipolar cell that goes up into the invagination, then that bipolar cell will respond. However, if transmitter is released at an invagination that's not innervated, then the bipolar cell, it'll be attenuated by diffusion distance and not activate the receptors here, and the bipolar cell will fail to respond. So the idea is that the bipolar cell should be sampling a constant fraction of the cone's vesicular output. And that's what we're looking for, and indeed, that's what we see in this model. So let me just walk you through the experiment very quickly. We record simultaneously from the cone and the bipolar cell. We give a maximal stimulus first to gauge the quality of the recording. This is indeed a large postsynaptic EPSC from the CB2 bipolar cell, and also a very large response from the cone. Then we dial the stimulus back to a much lower level, here giving about 80 consecutive stimuli and recording the responses. And I've grouped these responses in a helpful way to understand what's going on during this epoch of stimulation. The first responses I want to look at are the failures to respond. The black here shows the cone photoreceptor. Failures to respond in the cone photoreceptor when we then pull out the simultaneous traces in the bipolar cell, there's also a failure to respond here. This is good for us. If there was a failure here and a success here, then our method wouldn't be very good. Our transporters wouldn't be very good at telling us about cone release, but we find that that's almost never the case. Whenever we see something in the bipolar cell, there was always an event in the cone. The next sorts of events we look at are successes in the cone photoreceptor, detections of released glutamate, accompanied by failures in the cone. And this is consistent with the idea that at least some of the sites in the cone photoreceptor do not receive innervation. There is transmitter release from the cone, but the bipolar cell is failing to respond. The caveat here is that we hope that the bipolar cell is indeed sensitive at the single quantal level, and we can see that it is by isolating several very stereotyped traces at the quantal level, showing the single quantal response in the cone and the simultaneous single quantal response in the bipolar cell. And finally, we can see in this is a CB2 cell. A CB2 cell makes invaginating contacts and samples. We think from a fixed fraction of the invaginations and ought to see a fixed fraction of the cone signal. One of the ways we look at this, beyond just looking at the individual traces, is to plot for each event, to make a scatter plot of the peak CB2 cell EPSC versus the peak cone EPSC. What you can see in this scatter plot is, are we okay? Let's go to chat.
Yeah, it should be okay. Sorry.
Okay, so you can hear me. All right. What we can see in the scatter plot is that there's a linear fit which goes through the origin, which is consistent with the idea of that we're sampling a constant fraction of the cone output, irrespective of the size of the cone output. And then because of the behavior of the well-regulated behavior, both of the cone response and the bipolar cell response, we can actually convert from CB2 EPSC peak to bipolar cell quanta, the number of quanta which give rise to this peak response, for instance, in this case, 1. And we can also convert from cone amplitude to cone quanta, staying within the linear range of the cone transporter. And I neglected to mention that cone transporters saturate when more than 15 vesicles are released at the synapse. They're a very sensitive measure. We can convert this slope to bipolar cell versus cone quanta. And when you do that, the slope equals the fraction of sites sampled in the cone photoreceptor. So in this particular case, the cone is sampling 65% of the bipolar cells, release sites. Now, this method works very well for CB2 cells, but for CB1 and CB3 cells, their events are very difficult to characterize as falling into quanta. We don't want to make that assumption, and it's really not apparent how to do that. So in order to quantitate and deal with the model and deal with the responses of CB2 and CB3 cells, we developed a different way to plot the data. This data still quantifies the number or the quantal content of the cone event, but just as is there a success or a failure in the bipolar cell. It's a binary calculation, a binary observation. And the idea behind it is explained as follows. We give a train of pulses to the cone photoreceptor, and then we, at each response to the cone, we determine how many quanta are contained in that response. And we go through all the responses, and we look at the cases where the cone detected 1 quanta. And then we look at the bipolar cell and calculate the percentage of time the bipolar cell responded or failed to respond. And so, and then we go and we find the cases of 2 quanta and calculated the fraction of responses and so on. And so we don't have to know for the bipolar cell how many quanta are contained in the response. All we need to know is the failure or success. And the data from this particular cell is plotted here in red. There's about a 20% failure rate when 1 quantum is released, a 0% failure rate when 2 quanta is released. This is data from a different cell. The linear fits come from a model, and the model is very simple. In this model, we have 20 release sites, here shown in red, and a bipolar cell samples from a fixed number of the release sites. Here I'm showing you 6 release sites. And we make up a rule, and in this case the rule is that whenever a vesicle is released in an occupied site, a single vesicle, the bipolar cell will report a success. That can occur in any of the sites, and it's a success. If a vesicle is released outside of that, it's a failure. So in fact, and I've modeled this with 5 contacts, not 6, sorry. For 1 vesicle released in this configuration, there'll be failures 3/4 of the time. 3/4 of the time, randomly a vesicle will be released in one of the sites that's not innervated. If we randomly release 2 vesicles, there'll be failures 9/16. If we randomly release 3 vesicles, there'll be a complete failure to have any vesicle released under these contacts 27/64 of the time. So this is a rather simple statistical calculation. It's used to determine these lines, which are then lines of the probability of failures, calculated for each number of contacts. Here, 4, 6, 7, 11, or 16. And I fit the data points with the closest line. And you can see 16 contacts is roughly 80%, is 80% of 20. So by this method, we're seeing 80% sampling. It turns out that the size of this response is not an outlier. It's well within the size of the larger responses we see, suggesting that a good portion of the time, the CB2 cell is sampling from most of the invaginations in the cone synapse. And so this is a case then of comprehensive or near comprehensive sampling, or at least the suggestion of it. We're using super resolution to actually count the number of contacts, but I don't have images for the CB2 cell that I can show just yet, but I do have images for the other cell types, and we'll get to that in a moment. I now want to turn to the CB3 cell, and the CB3 cell is an important test case. This is a cell that makes multiple basal contacts, and they're very similar off bipolar cells to this in the primate and mouse retina. And the question is, do these basal contacts sample from all of the release sites? And the answer is going to be yes, they do. And so let me show you some of the data which supports this conclusion. Very similar to the experiment done previously, recording from a cone in a bipolar cell. This is an identified CB3A type of bipolar cell. We apply a series of brief depolarizations to the cone, here at 2 different voltage levels, evoking cone responses and simultaneous bipolar cell EPSC responses. The traces are color-coded according to their position in the sequence so that you can see corresponding traces in the cone and the bipolar cell, and also so that you can see that there's no systematic trend, that responses are truly bouncing up and down and are fairly stationary during these stimuli. And what jumps out immediately is that even though the cone responses are about double the size with the larger stimulus, the bipolar cell responses are more than double the size. And this is reflected in the scatter plot, where we see the green and magenta points shown here, as well as other data points from different runs. And this data is not well fit with the straight line, but much better fit with a curved line that starts at the origin and then ascends within the linear range of detection of the cone transporters. These responses are quite large. This is only a portion of the range. And if we were to trace this all the way to the peak, it would be off the top. But again, that is due to the fact that the transporter saturates after about 15 or so events. We fit these types of curves with a power law function. Here the exponent is 1.7. The average exponent is 2.3. So there is a substantial nonlinearity in the responses at these synapses. The responses are small or absent at low levels of release, in some cases absent, but more reliable at higher levels of cone release. It's a little bit more informative to look at our failure plots. And these failure plots suggest very strongly that the cone is sampling from nearly all of the release sites. The bipolar cell is sampling from nearly all of the release sites in the cone. Here shows a plot for this piece of data. At 1 vesicle released, these show the responses. There are 6 failures and 3 successes, giving us a point here. At 2 vesicles released, we see a similar point, but by the time the cone terminal is consistently releasing 3 vesicles, they're all successes to detect. And the fact that the cone photoreceptor is releasing only 3 vesicles suggests that there is fairly comprehensive sampling. And we'll model this in a moment and actually put some numbers on that. For comparison, I've taken the curve from the invaginating model, which starts up here at 5 out of 20 contacts sampled, similar to where this data starts, and then curves downward, but it's a very poor fit for the data in the CB3 cell. We're going to have to resort to a slightly different model to model what's going on here. This just shows some aggregate data showing that this decline to complete successes routinely occurs for this cell type when very few vesicles are released. Yet we have the paradox that when a single vesicle is consistently released, these cells only respond very rarely. There's a fairly high failure rate. Well, how do we incorporate that into a model? Well, we've changed the model a little bit. Instead of 20 release sites for symmetry, we now have 19 release sites. And these show the potential detection sites for the postsynaptic bipolar cell, which can fill either 1 or 2, all the way up to 24 sites, depending upon how we set the model. And in this model, we can propose a simple rule that a contact must detect 2 simultaneous vesicles being released in order to respond. And a simultaneous contact can occur, for instance, at this site if these 2 sites release vesicles, if these 2 sites release vesicles, or if these 2 sites release vesicles. And that can be a similar relationship for all of the contact sites. And so when we add such a rule to our model and plot the curves, what we see is very instructive to start by looking at this red data, is that we have to change the model a little, change the rule a little to 3 simultaneous releases equal a report of a detection success. And here, using this Basel III rule, we can fit the data with 22 contacts. For these more rapid declines, we need the Basel II model, shown here, 2 vesicles equal a response. And in some cases, and in all cases, it's very comprehensive sampling, 24 out of 24 sites. In this case, it's 19 sites. And in some cases, we have to resort to a hybrid model of the 2 models with single vesicle detection. A certain fraction of the single vesicles are detected and produce a response. But taken as a whole, this data and the modeling suggests that the CB2 cells transit from a region where a substantial number of failures are occurring due to a region of no failures due to very comprehensive sampling and a rule that you need to have 2 vesicles fusing at once in order to produce a response with certain compromises or adjustments to that rule. It supports complete sampling of the ribbon. We've used STED imaging to look at these synapses. The STED microscope is able to achieve super resolution imaging to break the diffraction barrier. A typical microscope illuminates a patch of data and measures fluorescence from that patch on the order of 250 nanometers in diameter. The STED microscope superimposes a depleting ring on that fluorescent patch such that the photons cannot be emitted toward the objective under the ring. Only the central photons can go up and be imaged by the objective. And depending upon the strength of the depleting donut, we can constrict this central point that can fluoresce up to a very small diameter, thereby achieving super resolution. There's also an approach to not only creating a donut, but lobes above and below on the z-axis that also deplete release, so that we can image just from a point right in the middle. And using this 3D STED, we can scan through our tissue in very much the similar fashion through which we can scan through an X, Y, and Z stack in the confocal microscope. This shows an old image from Wei Li's work, probably the best that we or anyone else can do at the cone to CB3 cell synapse. This shows a STED image of a GFP-filled CB3A bipolar cell. You can make out a number, you'll have to trust me, a number of cone contacts here, but I want to zoom in on a single contact, and that's shown here, and we can interpret this image as follows. There's a dendrite that comes up, it thickens, and it gives rise to these bushy structures, the contacts that are postsynaptic to the cone photoreceptor. There is a process here which crosses right underneath the cone terminal, which is contained in this collapsed stack. CB3 cells express GluK1 receptors at their synapses, and indeed, we can see these synapses here are painted with the GluK1 receptors. And I'll show you that again. And so this one cell, this is a single cone terminal spanning from here to here. This one CB3 bipolar cell sends processes up, and these processes co-localize with almost all of the clusters of GluK1 receptors. The remaining clusters probably belong to the other type of CB3 cell, the CB3B cell. There's a very minor contribution from CB1A and B cells, as we'll see in a moment. So super resolution supports the idea of this comprehensive sampling by this type of bipolar cell, which is common to both the mouse and the primate. And I can discuss the commonality at the end of the talk, if anyone's interested. CB1A cell has a slightly different type of sampling. It makes a single contact, basal contact with cone photoreceptors, at least in the confocal images. But here, the contact is much less sensitive, much more nonlinear, or it can be viewed in terms of having a threshold at low levels. This just shows the cone responses, and the cone responses fall into a very nice amplitude series. And only one of the responses here, shown in cyan, is accompanied by a response in the postsynaptic bipolar cell. And based on a number of factors, we suspect that this is a multivesicular response in which a lot of vesicles have been released just by chance nearby the contact of the CB1A bipolar cell. We can use these plots to determine a quantal size. Here, the quantal event size is 25 picoamps. We can fit these curves, which really cleave to the baseline at a very low level here, and only pick up later on, almost right before the nonlinearity in the cone transporter kicks in. And for these, the exponent that best fits in the power law fit is on the order of 3, consistent with a substantial threshold nonlinearity or substantial cooperativity of release. If we look at our failure plots, the failure plots look very different. Here, there are failures from 1, basically 2. This is probably just the chance coincidence of one event. 2, 3, 4. Finally, at about 5 quanta released, you begin to see a substantial number of successes. These are other examples of synapses. These synapses are very insensitive, high failure rates up to about 4 to 6 quanta released, and then a gradual decay down to a level where they respond 100% of the time. If we take our model and concoct rules to make curves to fit these synapses, what we see are rules similar to 3 vesicles required in order to produce an event, 3 vesicles detected by a single contact, and this gradual decline is consistent with 13 out of 24 contacts, a portion of the contacts. In these cases, the rules are 5 vesicles before saying I detected it, 3 vesicles and 4 vesicles. And in these cases, the model is consistent with effectively a large number of contacts. But the bottom line is that the CB1 cell contact is much less sensitive to cone release, yet at the same time appears to sample a significant fraction of that release. And that can occur if the cell is only responding at higher levels of release when most of the invaginations are contributing. We'll get to the mechanism of that in a moment. This just shows some super resolution images. Our old image, now the new images showing that instead of a single contact, each of these kind of fat connections beneath the terminal puts out 2 or more little processes, in this case 4 or so processes. But still, by and large, the same pattern is preserved. This is the extent of the entire cone terminal, as shown by GluK1 staining. The CB1 cell, its processes are only localized to a portion of the terminal. Okay, so I want to switch gears just a little bit to talk about receptor differences, because for a number of reasons, it appears that the CB1A cell is less sensitive to glutamate release. Even though it makes fewer contacts, it's even more insensitive than we suspect compared to the CB3 cell. And we're also interested in how the CB3 cell, with its basal location, is able to detect and respond to glutamate released at distant invaginations. Are those receptors sensitive, and are the CB1A cell receptors insensitive? And so the technique for characterizing the receptors involves pulling what are called somatic or nucleated patches out of the retina and bringing the soma over to a rapid perfusion device. And these perfusion devices can apply glutamate very rapidly, allowing us to capture receptor activation and also the very quick receptor desensitization. You just can't see that by applying glutamate within the slice. It takes about 20 minutes. You have to be very patient to pull a soma out of the bipolar cell. And I just want to show you a video of what that looks like. You patch onto the bipolar cell in the slice, you make a very tight seal, and then you gradually pull the bipolar cell away. And if you're very lucky, the axon and dendrites snap back, and then you pull the soma over to the rapid perfusion device. The soma is on the end of the pipette tip. These devices consist of side-by-side flows out of solution with a nice straight septum between the two pipes. Solution here control, here glutamate containing, flows continuously. And we move this interface very quickly over the cell by mounting the perfusion pipettes on a laterally moving piezoelectric device. And we can get sub-millisecond transitions in that case. So the data then looks as follows. We pull the somas off. The somas have a substantial amount of receptors, particularly so for the CB3 cells shown here. And one of the nice features of this technique is that we can fill our patch pipette with dye. And when the axon and dendrites snap back, they're filled with dye, and we can image the cells and determine precisely what type of bipolar cell, whose soma we have very rudely pulled out of the retina. In this case, this is a CB3A cell. In this case, a CB1A cell. During an experiment, we can perfuse this barrel of the pipette with different concentrations of glutamate and obtain concentration response curves. These concentration response curves are shown here for the CB3 cell on a slow and on a fast time base, and similar for the CB1A cell on a slow and a fast time base. And the take-home message is that the receptor affinities are different. For these two cells, a plot of normalized amplitude response versus glutamate concentration has a difference in EC50 of about threefold. In the entire sample, that difference is a little bit more than fourfold. The glutamate receptors, the kainate receptors on the CB3 cells have an EC50 of about 330 micromolar, and those on the CB1A cells are considerably higher at 1.4 millimolar, very high for ionotropic glutamate receptor. And we wonder what sort of subunits give rise to such a low affinity for glutamate. The ends for the receptors are very similar. They're on the order of 1. And I don't have time to go into why that makes sense for the synapse not to have different ends for the two types of receptors, but it affords a simplicity. We're interested also in detection at the threshold. So in this experiment, I applied lower concentrations of glutamate, an initial high concentration to test the peak response of the receptor. But now we're thinking in terms of basal context, where the glutamate concentration is estimated to be about 20 micromolar. Do the two types of receptors respond at 20 micromolar concentrations? Here you can see the effect of liposuction in these confocal images. The somas have been removed, leaving the cells behind. This is the chat band, helps us identify the CB3A cell, CB1A cell. And the take-home message is that there's a substantial response in the CB3A cell at low concentrations of glutamate, 25 micromolar, similar to those estimated to be found between invaginations. So CB3A cells can respond at the single vesicle level, whereas CB1A cell responses are exceedingly small at these levels, and for all intents and purposes, absent. They're down by about 200 to 300-fold relative to the peak response, as shown by this log-log plot. We've done a lot more with this data on the receptor properties in collaboration with Drs. Futagi and Kitano. We've modeled the properties of the CB1, and we're in the process of modeling the properties of the CB3 cell receptor. And these models allow us to explore features of processing at the synapse, which would be inaccessible to patch recordings. Okay. So I want to summarize our results as follows. Now, this is not an anatomist's view of the cone photoreceptor synapse. This is how a physiologist looks at the cone photoreceptor synapse. We've erased all of those wonderful tiling basal contacts and replaced them with functional contacts. And the main point here is that the CB1A cell makes roughly 3 or 4 contacts right in the center of the cone terminal. But more importantly, from the point of view of sampling, the CB3 cells sample from every invagination. And we've symbolized that by having contacts above and below each invagination for each type each of the two types of CB3 cells making contact. In addition, I've drawn the CB2 cell making its contacts in the invaginating position, sampling from almost all of the contacts. To continue with the graphical summary, what we think of combining receptor affinity and receptor sampling is the following type of sampling. The CB2 cell makes its contacts close to invaginations, and here I've inverted the cone synapse upside down like a cup, so that when there is a small amount of transmitter release, these contacts can detect and respond to that transmitter release. The CB3 cell is located basally outside of these invaginations. It requires a little bit more release to produce a response, but the CB3 cell is fairly sensitive and integrates responses, as does the CB2 cell from the different ribbons. And the CB1A cell works according to a different principle. Here, its responses are more tidal in nature. It has a less sensitive glutamate receptor, which responds chiefly when glutamate begins to be released from multiple sites at the terminal, begins to fill up the space under the cone terminal. So when I mentioned about tidal release and just sampling the tide in one position, the CB1A cell seems to read out for most of the contacts of the cone terminal, but only under higher release conditions. And this just repeats the summary I've shown here, integrating the idea that the different bipolar cells have different response ranges during the various phases of cone transmitter release, from relatively low variations in transmitter release detected by the CB2 cell in the invagination to moderate ranges of transmitter release at light off, and also during the sustained response detected by the CB3 cell to the extreme concentrations of glutamate detected by the CB1 cell. And there's quite a bit of overlap in these ranges. So finally, I want to take a few moments, just very briefly, to tell you about our recent work with transcriptomics and proteomics to try and understand. The long-term goal is to try and understand the molecular basis giving rise to these different contacts. The immediate goal is to try and understand what different subunits, what different kainate receptor subunits can account for the difference in the CB1 and CB3 cell receptors, with particular emphasis on trying to understand this very low affinity CB1 cell receptor. The glutamate channel is a tetramer of subunits. This is not my work here. This is taken from Wikipedia. And these tetramers can be comprised of 5 different subunits. GluK1, and I have these asterisks here because GluK1 predominates at the cone photoreceptor synapse by antibody labeling and by in situ hybridization in these off-bipolar cells. GluK2, some people feel this is present at cone photoreceptor synapses. And GluK3 is another subunit, and this is a very low affinity subunit. It's very uncommon to find GluK3 in postsynaptic receptors, where normally the glutamate concentrations are too low to activate the receptors. All these 3 subunits as homomers and also as heteromers can form functional channels. GluK4 is not found in the retina. GluK5 doesn't function as a channel on its own, but in combination with the other subunits can adjust its properties. And we know of a number of auxiliary subunits, the NETOs, NETO1 and NETO2, which can change the properties of these receptors. So we're interested in exploring this with transcriptomics. I don't have time to give you the whole story, but we dissociated the ground squirrel, adult ground squirrel retina. We ran downstairs to the 10x genomics machine, and we injected the cells into the machine. And several months later, we got the mRNA readouts for the individual cells in our sample. And then we focused in particular on the bipolar cells. And we did this by looking at the original clusters that express OTX2. OTX2 is expressed by all bipolar cells, but also by the photoreceptors in the ground squirrel retina. But we were able to eliminate the photoreceptor clusters by observing their expression of green opsin, blue opsin, rhodopsin, and arrestin-3. This just shows a t-SNE plot after PCA analysis of the OTX2 positive clusters. And this shows the OTX2 labeling in these clusters. We can see that indeed we isolated some good clusters. We then used the antibody markers for the different types of bipolar cells to try and identify which clusters these are based on the message expression. PCP2 lights up 3 clusters. PC2 is expressed by on bipolar cells. And by using other on bipolar cell markers, we could establish the identity of these clusters above the line in our plot, and we can actually account for the major types of ground squirrel on bipolar cells. For us, minor types are rod bipolars and blue cone bipolars, and we haven't been able to identify those with certainty. The off bipolar cells are below in this plot. Off bipolar cells, it turns out, almost uniformly express GRIC1, and that's shown here. There are 2 minor exceptions, a FESF1 positive cluster. These are the so-called gloomy cells in the mouse, which in the mouse don't reach up to contact cone photoreceptors. And then there's another cluster that's very weak for GluK1. And it's surprising that it contains GluK1 message at all, because these are the CB2 cells by CABP5 labeling. And these cells express entirely AMPA receptors at their synapses. Okay, so through the same process, we're able to identify the cell groupings of the off bipolar cells, not as elegant in spite of our best efforts at clustering as for the on bipolar cells. The broad expression of GluK1, and now I want to look at GRIC2, and I'll do that again, focus here. You can see that GRIC2 is also expressed by a portion of the CB1 cells, CB1 cell clusters, suggesting co-expression of receptors and providing at least a preliminary clue that receptor diversity could underlie the different properties in the different receptors of the bipolar, off bipolar cell types. We then looked at NET01, and NET01 is broadly distributed, but also there's a pattern to the distribution, less in some cells, more in others. We're also considering very strongly the contribution of NET01 to the kainate differences in kainate receptor responses. Finally, for the proteomics, we recently succeeded, we have a very nice antibody to the GluK1 subunit, which is only expressed by bipolar cells. We ground the retina up, we took the membrane fraction, we pulled down GluK1 receptors and whatever comes with it, and then we ran what we got out of that pull down through the mass spec, and we were able to identify a number of subunits and proteins that are associated with the GluK1 subunit. That includes GluK1 itself at a very high level, GluK2 or GRIC2, NET01, GRIC3, and GRIC5. The GRIC1 is at a very high level compared to the GRIC2, and how we interpret this data is that it's very likely that most of bipolar cells, probably the CB3 by volume of contacts, express predominantly GRIC1 receptors. And then there's a subset of bipolar cells in which GRIC1 is expressed in combination with GRIC2, potentially NET01, GRIC3, and/or GRIC5. And so this is just work in progress. I wanted to give you a feel for where we're going here. So I want to give credit to my various co-authors. I'm responsible for all of the physiology, but I've had tremendous input from my collaborators, Daiki Futagi, who's done fantastic modeling, and Dr. Kitano, who has helped guide Daiki through this modeling. The transcriptome work, Junshi helped me with the dissociation, and my daughter helped with the analysis of the data. She mastered Seurat, the program Seurat for this purpose. The proteomics was done by Latha Lee, who's a technician in the lab, in collaboration with Jeff Savis, who that's upstairs, not downstairs. We ran upstairs and used his mass spec. And then the STED imaging is done in collaboration with Eric Schwartz, who's really had a major role in developing the approach. Vitas Bendakis, who is the head of the imaging facility at the University of Chicago, and Jun Shi in the lab, who has been very innovative in developing our embedding technique. And I want to give credit for the funding agency as well as for the Center for Systems Vision Science. Thank you. It's a little bit eerie to give a talk with no feedback, but I hope I can receive and respond to your questions now.
Thank you very much, Steve. And it is so great talk. And also, like, I'm so impressed with your beautiful physiology work as well as 3D super resolution work as usual. Thank you very much. And then, like, now, this talk is open for discussion. And like, are there any questions? 今このシステム上の問題で、ご質問いただける場合には、チャットの方から、ご記入いただけますとありがたいなというふうに思っております。So actually, my question is probably very primitive, but I'm wondering the three cone synapses, how do they contribute for the visual formation at last?
So, I mean, that's a very good question. I think you're asking, which types of ganglion cells do they contact in their inner retina, because if you know which type of ganglion cells, then we can say what type of information those ganglion cells encode. So I don't have that information in the ground scroll. We know that the CB2 cell, by its nature, is an achromatic bipolar cell. It contacts both the green cones, the blue cones, and the rods, as it turns out. And it's a very fast cell. It's a very large cell. It has fast AMP receptors, and it makes invaginating contacts. And so we've kind of pegged that cell as a fast achromatic pathway. The other cell types I'm not so sure about, but maybe this will help. This is a slide that you and I have spoken about. Can you see the slide here?
No, I don't see.
All right.
Would you share?
Let me share. Do you see it now?
Yes.
Okay. And so this is kind of a tentative mapping, and maybe this will help people understand a little bit about what sort of signals are being carried. I can't really tell you at the ganglion cell level, but the CB3A and B cells I've been talking about that strongly express KN8 receptors and that sample broadly at the cone synapse appear to be analogous in many ways to the type 3B and type 4 in mouse and the DB3B and DB2 in the primate. So for those people, and there are probably one or two in the audience, who can more readily relate the bipolar cells in the mouse and the primate functionally to ganglion cell types, this may be helpful. I can't really do that myself. And by the same token, the CB1 cells appear to have parallels among the flat midget bipolar and DB1 cell in the macaque and type 1 and 2 in the mouse. And this AMP receptor expressing fast cell in the ground squirrel is very similar to these KN8 receptor expressing DB3 or 3A types in the mouse and primate. So maybe that will give people an idea of the function. We're not completely at sea, but I can't quite hook this up all of vision just yet. So I'm going to stop sharing. Okay.
Yeah. Yes, thank you. Thank you very much.
And you know that. You've seen that diagram because we put that in a grant.
I mean, like, I'm wondering if you are seeing Dr. Tachibana's question.
Okay. So I see the question. Excellent talk. I would like to know the geometry of EAT5 relative to the vesicle fusion site because the anion current may depend on the distance between EAT5 and the glutamate release site. Well, I would really like to know that too. And we're just in the process of doing those super resolution images. I showed super resolution images earlier in the talk when I showed the two cone synapses, but they just weren't of the quality to really address the issue that you're asking me to address. In this technique, we don't have good ways to fill the cone. Whenever we do that, the signal comes out way too bright and drowns out everything else. So we've really biased the super resolution technique to look at different proteins at the synapse. So for instance, for the invagination, we use ribeye, GluR4, which is also expressed by horizontal cells, and the CB2 bipolar cell, SAP102, which is a horizontal cell protein. And we use these various proteins to kind of peg the localization of the invagination. And we were very interested. We also use PSD95, which provides a beautiful outline of the terminal. I could go back to the first slide, and I will go back to the first slide in a moment to show you what that looks like, the wallpaper slide. But the point is that we really want to look at GluK1 and EAT5 and the relationship to each other and the relationship between those two and the mouth of the invagination. And what we hope to see, and what we think is going on, even from preliminary images, is that the GluK1, that the EAT5 is not in invaginations, that it's located basally, and that transmitter has to diffuse over the EAT5 in order to make it to many basal contacts. We haven't seen a precise colocalization between the CB3 cells and EAT5. So it's not like there's a cluster of EAT5 below the CB3 cell, but we do see clusters that are in between the GluK1 receptors. So let's see. So the answer to the question is that the super resolution supports the idea that EAT5 is very close to the synapses. It's not inside the invagination, but it's there. And hopefully by the next talk, it's really a priority of ours to have much better images of that.
Thank you very much.
Is that good?
Yes, great.
Okay.
Okay, then actually, Dr. Tsukamoto have a question.
Okay.
And please let me read.
Okay, please do.
So in the macaque retina, there are three positional classes of off-cone bipolar cells. One class, which has basal contacts far from invagination, has also contacts with rods. I wonder if CB1A has contacts with rods as well. It's a question from Dr. Tsukamoto.
Yeah, so that's a very good question. So the CB1A cell does not contact rods. It's only the CB2 cell. My feeling, and probably you share this, is that there's a lot of variation in the patterns of bipolar cell to cone contacts between species. Which bipolar cells contact the blue cones, which bipolar cells occasionally contact rods, which contact the green cones. That's probably something that strikes me as being very sensitive to the visual environment of the animal. Yes, the CB1A cell doesn't make contacts with rods, and probably there is not a direct analogy then to that particular type of bipolar cell, but there could be a similarity in other respects in that case. I hesitate. You know, when I first started diagramming the synapse, I diagrammed the synapse as kind of a bullseye, where the CB2 bipolar cell was at the center of the ribbon, the center of the bullseye. The CB3 cell was around next in the rim, and the CB1A was around further than that. Physiologically, we have no information for such a precise organization, and probably even with super resolution microscopy, it'd be very difficult for us to obtain that. So I should go back and look at your EM study again and pay a little bit more attention to that. Okay?
That's great. Yes. Thank you.
Good.
Thank you very much. Thank you very much. It's great.
Mhm. Oh, thanks.
So,
Any more questions? I'm sorry I'm not able to share food after. That was my favorite part. We all go and have, a bento box.
but now, like your time is 1 a.m.
Yeah, my
in
time
Chicago.
is late. Um, and so it's probably too late for me to eat anything.
Yeah. Thank you
But
very
that's
much.
fine. That's fine.
Yeah.
Good for me. It's good for me. So I hope that you'll enjoy yourselves.
Yes, this is the best time for us. So,
Yeah.
thank you. Thank you very much. So I think now, like, is
I
um,
can wait a moment. I'm not in a rush to go anywhere. I can wait a moment in case anyone has an additional question. Um, so yeah. It's up to you.
now I want to close the first seminar. Thank you very much for all your participation, and I hope we would see you again as next talk in the future.